While genetic engineering in developed countries have been used to make foods easier to farm, more visually appealing, and increased its quality of taste, in developing countries, GMO foods have been used to solve severe nutrient deficiencies. One such example is how golden rice is used to solve vitamin A deficiency, or VAD. This is how biotechnology achieved it, along with how this food helps the body and even backlash it received. In less developed countries, around 500,000 children go blind every year due to vitamin A deficiency. Sadly, more than half of these suffering kids lose their lives to this nasty form of malnutrition. This vitamin A deficiency is one of the most prevalent in the developing nations of Bangladesh and the Philippines. Interestingly, like many other countries in their region, their population is highly dependent on rice for sustenance and survival. Scientists of the Rockefeller Center have engineered a momentous golden rice variety. This rice is such a timely invention as it has proven to combat vitamin A deficiency quite proficiently. Scientists predict that a singular cup of rice a day is enough to delay or prevent blindness in children and adults. This rice utilizes the power of introduced beta-carotene, which is the main ingredient in how this rice is used to combat vitamin A deficiency. Beta-carotene is a precursor to and synthesizes the active vitamin A, or retinol. Beta-carotene is also an effective antioxidant. Antioxidants are substances which prevent or delay cell damage and deterioration. As well as this, they can prevent inflammation in the body. Due to all of the reasons above, scientists altered the genetic sequencing of rice crops to incorporate a beta-carotene gene. So how exactly was golden rice created? What was the science behind it? How exactly was its genetic sequencing altered? Golden rice falls under the GMO category of transgenic rice. This is because it is modified by including genomes from various other organisms. The rice variant was engineered by adjusting the rice genome to incorporate a gene from the wild daffodil plant, Narcissus pseudonarcissus, which constructs the enzyme phytoene synthase, and a type of bacteria called Erwina uridivora, which creates the enzyme phytoene desaturase. These two enzymes are crucial in the functioning of golden rice. Their role is to enhance beta-carotene accumulation in the endosperm of the plant. The fact that the beta-carotene accumulation is high in the endosperm is important, as this is the edible part of the rice plant. So how exactly does the extraction of the beta-carotene gene and its input in the rice crop occur? The beta-carotene gene in the daffodil plant is cut using restriction enzymes. Rice cells contain DNA rings called plasmids suspended in their cytoplasm. A section of this plasmid is cut using the same restriction enzymes, and since both the plasmid and the beta-carotene gene on the daffodil and the rice are cut by the same restriction enzyme, their ends are complementary. The complementary bases bond together and produce an integrated plasmid, creating a transgenic golden rice organism. CRISPR is inspired by an autoimmune response to viruses used by bacteria. When the bacterium becomes aware of the presence of virus DNA, it constructs two short strands of RNA. One of these emulates the template strand of the actual virus. The two RNA strands, together with a protein called Cas9, form a complex. This Cas9 protein is instrumental in the DNA editing procedure, as it is actually a type of nuclease enzyme, which means that it can cut DNA. After the guide RNA, which is the copy of the viral DNA template strand, bonds to the other strand of DNA, the Cas9 in the bacterium's RNA complex cuts the viral DNA away from the bacterium, disabling the virus. This response involving the cutting of DNA and more specifically the ability of Cas9 protein to isolate genes is what allows for genetic engineering and therefore golden rice. So this is how bacterium does it, but how does it work in the case of golden rice? The bacterium's RNA Cas9 complex is utilized to pry into the nucleus of a living cell. After finding the genetic coding in the nucleus and the DNA, the complex latches onto a small segment of the sequence called the protospacer adjacent motif, otherwise known as the PAM or PAM. The Cas9 helps to unzip the DNA, replacing the template strand with its target RNA or guide RNA copy. When the DNA strand is successfully unzipped and replaced, the Cas9 enzyme cuts the DNA. After the DNA is cut, the cell immediately tries to respond and rejoin the DNA, but this process is error-prone. These errors can result in mutations leading to an abnormal expression of the gene or even no expression of the gene whatsoever. This is an important part, as now scientists can understand the role and responsibility of the gene that was cut. The DNA in the cell can be repaired by using a healthy and functional and desired segment of cut DNA using CRISPR. This can resolve the mutation as this new functional gene will cut the mutation and instead join the sequence due to base complementation. There is widespread opposition to the advent of golden rice due to reasons such as more natural alternatives, cultural rigidity, unfair competition with large corporations, and stigma against genetic engineering and modification. Some organizations opposed the golden rice Some organizations opposed to golden rice include Friends of the Earth organization in the USA and the Masipag organization from the Philippines. Their arguments include how there is already a UN initiative combating VAD which could increase the survival rate from 12% to 24%, and how this could be employed to divert attention from the UN initiative, allowing the Golden Rice Project to earn a profit. It can also be argued that golden rice may contain too little beta-carotene, 1.6 milligrams of beta-carotene per gram of rice, whereas the requirement is 10 times that. When the debate gets too intense, as with many subtopics under the GMO foods category, this happens.
In August of 2013, hundreds of them stormed a Philippines government test field planted with golden rice.
Mga pananim partikular ang golden rice.
They uprooted and destroyed all the plants.
While it is clear that the motives behind golden rice are incredibly thoughtful, and despite the opposition that is trying to block its tracks, it is very clear that golden rice still has a future ahead filled with new scientific discovery and innovation.